Hello, my name is Beatrice Setnik, and I'm the Chief Scientific Officer at Altasciences. Today we'll be getting at the heart of the matter, the considerations for early versus late QT prolongation testing. Some drugs that are not designed with cardiovascular indications may have effects on the heart, including delayed cardiac repolarization and increased risk for dangerous cardiac arrhythmias, including torsade de pointes. The E14 guideline of the International Conference on Harmonization states that specifically designed thorough QT or TQT studies are a crucial element for the clinical assessment of the potential cardiac risks of any drug. Drug-induced QT prolongation is one of the leading factors of regulatory approval delays and drug removals from the market. The TQT study is typically conducted during phases 2 or 3 of drug development, a time where there is more data available regarding the pharmacokinetic profile, maximum tolerated dose, and proposed therapeutic dose. The objectives of these studies are to determine if the drug-induced effect on the heart rate corrected QT interval upper bound of the 95% one-sided confidence interval excludes 10 milliseconds, or the threshold of regulatory concern. The TQT studies require large sample sizes and include both an active control, such as moxifloxacin, for assay sensitivity, as well as the placebo. The Cardiovascular Renal Division of the FDA will consider the determination of clinical QT prolongation with the use of concentration effect modeling. This enables testing to be conducted early in clinical development, with inclusion as early as the first-in-human or FIH studies. Criteria must be aligned with the E14 guidance. The single ascending dose, abbreviated SAD, and multiple ascending dose, abbreviated MAD, study provides examination of a wide dose range given both as single and multiple doses. These studies cover a large variation in drug exposure and can serve as useful correlates for concentration effect modeling. Since these studies are principally focused on general safety evaluations, modifications need to be considered in order to include intensive cardiac monitoring and exclusion of subjects with QT irregularities. The QT evaluation implemented is one with the same rigor as that in the traditional TQT study. Early assessment requires the use of modeling. The concentration effects models must be predefined and include data over a wide range of plasma concentrations. This range should cover subtherapeutic to supratherapeutic levels that are at least 3 to 5 times higher than the expected therapeutic concentrations. If the early QT assessment is scientifically rigorous and adequately designed, there may be the potential to waive or eliminate the need for the traditional TQT study for some drugs. In the event that the early assessment shows a signal, thus requiring a TQT evaluation, the data obtained can be utilized to inform the TQT study design as well as the sample size. Early QT assessment allows for early de-risking of a compound, which adds to the safety and data package of a drug, informative to the decisions affecting drug development strategy. Since early examination is both cost and time effective, there are additional costs and time savings if a TQT waiver is obtained. To defer costs, it is also possible to collect the data early and conduct analysis and modeling later in the drug development cycle. PK and PD modeling and simulations are utilized in the early assessment approach. In the concentration effect model, the pharmacokinetic PK data, concentration of the drug over time, is time matched with ECG data to create a measure of exposure and response. The model relies on exposure responses using the amount of drug as exposure and the QT interval as the response. A number of studies have been conducted to date with early assessment approach. An application for a TQT waiver can be submitted with valid data. If granted, a TQT study is not required. Factors that can affect FDA waiver approval depends on how much data is available at supratherapeutic doses, consideration of drug interactions, the method of ECG collection, for example, triplicate versus single readings, manual versus computerized readings, and whether or not there is an effect of the drug on the heart. There are several important limitations to early QT assessment. While many drugs can benefit from early QT evaluation, some drugs may not be good candidates for the TQT waiver strategy. In general, drugs that have either a positive HERG in preclinical development, an increase in heart rate, or other autonomic effect, require lengthy titrations to achieve therapeutic concentrations, or are generally long-acting, may not be suitable candidates. Currently, there are no published guidelines for determining exposure-response QT modeling. Current designs assume false negative and false positive rates will be minimal. Drugs that have a borderline effect may not be detected. Study bias must also be considered, including blinding status, where blinded ECG overread is recommended for unbiased QT analysis, as well as conditions of drug administration. For example, food can impact the QT interval with some clinical evidence that has a show of shortening of the QT of up to 27 milliseconds following a carbohydrate meal, an effect lasting up to 4 hours post-consumption. SAD-MAD studies typically have very small sample sizes for each cohort. Typically, these are conducted in cohorts generally ranging from 8 to 12 subjects, where approximately 3/4 of subjects are allocated to active treatment and the remaining are randomized to placebo. The placebo and active treatments are not administered in a crossover fashion, hence one does need to account for inter-individual variability. In order to be deemed an acceptable alternative to the regulators, the rate of false negatives in the QTc analysis must be low. Alternatively, if the methods present a higher likelihood of false positive results, then a thorough QT study may be advantageous. TQT studies are typically conducted as parallel or crossover studies, usually with a single doses that approximate steady-state plasma levels. Treatments will commonly include the investigational drug at both therapeutic and supratherapeutic dose, a positive control such as moxifloxacin, and placebo. The sample sizes are large, ranging from 50 to 60 subjects per treatment arm. The ECG evaluation typically consists of monitoring subjects at baseline for 24 hours pre-dose and at matching post-dose intervals and time points. Conducting a TQT study after phase 2 allows for the collection of informative data regarding the targeted therapeutic dose and delays the cost incurred for the conduct of the study only for compounds that clearly show potential for QT prolongation. At this stage of drug development, there's a clearer understanding of the pharmacokinetics of the parent and any active metabolites and their relationship to efficacy. This allows for a more informed design and investigation into QT effects. As the TQT studies are large, they are also costly. For this reason, early examinations that produce data that can support a waiver may be a reasonable option for some drugs. Another consideration is the required exposure of a large number of subjects to moxifloxacin, an agent with known QT prolongation effects that serves as a positive control in these studies. So in conclusion, examining the effects of a drug on QT prolongation can serve as a useful de-risking tool to potentially waive the need for a traditional thorough QT study. As you can see from this recent example, receipt of a waiver is possible but does require proper planning. While certain drugs may not be suitable for this approach, most drugs may benefit from early assessments, providing time and cost efficiency. In the event that a TQT study is required, ECG data collected early can be informative for study design considerations. Thank you for your attention. I'll be happy to respond to any questions.